So, now we talk about laboratory glassware. It is a collection of highly specialized equipment designed to measure, mix, heat, store, and transfer chemical during experiments. But this isn't ordinary glass. To survive the extreme environments of science, most lab glassware is crafted from premium borosilicate glass. This material is uniquely engineered to be highly heat and chemical resistant. It won't shatter when rapidly heat or cold, and it won't react with the volatile chemical inside it. This specialized construction is what ensures your laboratory work remains both safe and incredibly accurate. Now we talk about this TD and TC. When working in a lab, accuracy is everything. So that's why laboratory glassware is calibrated in two distinct ways. TD means to deliver and TC means to contain. Understanding the difference is the key to perfect measurements. As for TD, the glassware with this mark is calibrated to measure the exact volume of liquid that is dispensed from it. It accounts the tiny film of liquid that naturally clings to the glass wall after pouring. Because they are used for accurate liquid transfer, pipette and burettes are classic examples of TD glassware. You rely on them to deliver a precise amount of reagent directly into your reaction. While for TC, this type of glassware is calibrated to measure the exact volume of liquid held inside the vessel. If you pour the liquid out, you won't get the full amount because some will stay behind on the glass. On the other hand, volumetric flasks are designed as TC glassware. Their purpose is to hold exact volume, making them the ultimate tools for preparing precise standard solution from scratch. So this is pipettes. Its core function is simple but critical, which are to measure and transfer precise volumes of liquid with absolute accuracy. Whether dealing with milliliters or tiny fraction of a microliter, it ensures that every drop is accounted for without ever contaminating the instrument. Because of this precision, its application spans almost in every scientific field. You will find pipettes at the heart of molecular biology for DNA sequencing, in medical labs running diagnostic blood tests, and in chemical analysis creating exact solution. Types of pipettes and its function. Volumetric pipettes. Its function is to measure and transfer one fixed volume of liquid with very high accuracy. It is commonly used in preparing standard solution. Second is graduated pipettes. It has a graduation marks that allow different volume of liquid to be measured and transferred. It is suitable for general lab work. Third is posture pipettes or a dropper. It's usually being used to transfer small amount of liquid drop by drop, and it is not designed for accurate volume measurement. And the last one is micropipettes. It is used to measure and dispense very small volume, which is in microliter. It widely used in biology, microbiology, and molecular research.
Right. All right. Now I'm gonna talk about ways to calibrate a pipette. So for the first step is the preparation. We must weigh a dry aluminum flask with a rubber stopper or a weighing bottle with a glass cap. Second is the fill process, where we must fill the pipette with distilled water and carefully record the water's temperature. Third one is the transfer process, where we must deliver the water into the wave container using proper pipetting technique. Last but not least, we must quickly add stopper to the container to avoid evaporation loss. Then we wait to determine the wave of the water delivered.
So this is our sample of data that we get from our
pipette calibration experiment. So as you can see, the average actual volume of our pipette that we use is actually 25.001 milliliter. Alright, next I'm gonna introduce you to the next classway, which is burette. Alright, what are the functions of burette? It is designed for the accurate delivery for of a variable amount of solutions. The delivered volume can be read to the nearest 0.01 ml via interpolation, accurate to about plus minus 0.02 to 0.03 ml. Burette must be rinsed with the solution to be measured prior to use. So next is the application. When or where do we use burette? So it's commonly found in a laboratory, primarily used in a titration process, where a standard solution, titrant, is carefully added to a simple solution, analyte, until the chemical endpoint is reached. Alright, now I'm going to talk about different types of burette and their own functions. The first one is the conventional burette. It is common types of glassware found in a laboratory for primarily for standard titration. Second one is the microburette. It is designed for microtitration that require extreme precision to deliver small amount of liquid, often 10 ml to 5 ml or even less. The third one is the electronic burette. It is a digital display or electronic mechanisms to deliver liquid and helps eliminate human error like reading a meniscus. Last but not least is the wave burette. It is a mass-based titration useful for viscous liquid. So next is the calibration of a burette. As for the procedure, it is the same basic step as pipette calibration, but deliver and weight several different volumes. So what are the purpose? So the internal pore of a burette is not perfectly cylindrical and it can be slightly wavy. So calibrating at multiple increment account for volume variation above and below the nominal markings is needed.
Volumetric flask. Volumetric flask are precision glassware designed to hold an exact volume of liquid. Types include 10 milliliter, 25 milliliter, 50 milliliter, 100 milliliter, 250 milliliter, 500 milliliter, and 1000 milliliter sizes. Function used to prepare standard solution of precise concentrations, ensuring accuracy in titration, solution, and quantitative chemical analysis in laboratory settings. Function of volumetric flask and when and where to use it. The primary use, volumetric flask is designed for one specific purpose, which is preparing and measuring extremely precise volume of solution. When to use it, you use volumetric flask when you are making standard solution, which is a solution with an exact non-concentration, or when performing precise serial dilution. And where do we use volumetric flask? It belongs on the analytical bench. You would never use a volumetric flask for heating, boiling, or storing liquids for long term, as extreme temperature changes can warp the glass and ruin its precise calibration. Physical characteristics of volumetric flask. Firstly, the design. It is a flat bottom glass bulb with an elongated narrow neck. It comes fitted with a stopper, usually glass or plastic, to prevent evaporation and allow for vigorous shaking or mixing. Secondly, classes. Typically available in class A, which is highest precision used in analytical chemistry, and class B, lower precision, which is commonly used in educational labs. And lastly, calibration. It features a single precisely etched calibration mark, which is the meniscus line on the neck. It is calibrated to contain Tc, a specific volume of liquid at a specific temperature, which is usually 20 degrees Celsius. Volumetric flask calibration. Firstly, the preparation. We determine the mass of an empty clean volumetric flask, for example, 50 milliliter. Secondly, the fill. We fill the flask with distilled water exactly up to the calibration mark. Next, we clean it. Clean the neck to ensure no water droplets remain on the neck of the flask. Blot them carefully with tissue paper if present. And lastly, weigh and calculate. Record the water's temperature and reweigh the flask. Determine the exact volume by using the mass of the water, which is found by difference and the standard water density table.
And this is another of our sample data for volumetric calibration. So as you can see, the final actual volume of the volumetric flask after overall calculation is actually 49.76 milliliter.
Conclusion. In conclusion, the appropriate selection of and correct handling of laboratory glassware are critical to accuracy and dependability in scientific experimentation.
In the analytical process, very specific and specialized functions are built into high-precision instruments such as volumetric flasks, pipettes, and birettes. It is important to use each tool for the purpose it is designed for in order to reduce the chances of mistakes made by the system or by people. Proper handling and technique of this glassware will ensure the precision and reproducibility of data, thus protecting the overall integrity and validity of the final experimental results.